everyone, welcome back to AP Bio. We continue our unit on chemistry and the review of chemistry and leading into our discussion of biomolecules, we're going to look at how the biomolecules are linked together. So we're going to be looking at something called a condensation or dehydration reaction. So a condensation reaction, also commonly referred to as a dehydration synthesis, is a chemical reaction in which two molecules combine to form a larger molecule together with the loss of a molecule of water. And the key thing here to remember is when we look at them, there's going to be these functional groups that are going to be sticking off the end, and usually it'll be an H and an OH right next to each other, and you can see from that, that's where the water comes off. That's why it's called a condensation reaction, and in this case, we're making an amide bond, and two molecules have been linked together to make a larger molecule. So again, condensation or dehydration reaction, they're used to connect monomers. So we're going to get into the vocabulary a little bit in terms of the single pieces, and we're going to link those together with a covalent bond to make polymers. It always involves the loss of water. The cell expends energy to do it, and the big thing that we'll spend more time on later is they need enzymes. So whether it's a ligase, a sucrase, something, there's an enzyme that helps facilitate that reaction. With the general formula being that we're going to take individual monomers and we're going to link them together with the loss of water, and for every bond that's made, one molecule of water is released. So here's the kind of the general formula that we're going to follow, is that we're going to take individual monomers, so whether they're amino acids, monosaccharides, or whatever the individual pieces, and we're going to link them together to make a polymer. So a protein is a polymer formed of the monomers called amino acids. We can take the monomer of nucleotides and string them together to make the polymer, which is going to be a nucleic acid or DNA. So plastics also work the same way. So we would take the individual monomers, and here are the different colors, monomer 1 and monomer 2. And again, you're looking, this is what you need to be able to identify. There's that H and the OH that stick together that's going to lead to the release of water over here and the formation of a new bond that makes, in this case, the copolymer. So plastics are another example. We're not going to talk about them anymore because they're not biological, but condensation reactions do take place there with the release of water. And here's just kind of a quick cartoon diagram of it. So we've got a short polymer with 3 monomers that have already been linked together. We're going to add a 4th one to it with the release of water, and that's how our polymer, this is how our biomolecules are going to grow into tens or hundreds or sometimes even thousands of individual monomers linked together to make our new larger biomolecule or macromolecule. And now let's look at some specific examples. So when we talk about proteins, what we're going to be looking at is we're going to be looking at 2 amino acids that we're going to link together. And in this case, we've got our acid part that will have the hydroxyl, the OH, and then we've got our amine part, the H. And when those are put end to end, they line up perfectly for a condensation reaction to take place. Water's going to be released, and in this case right here, we're going to make what's called a peptide bond. So that's the specific type of condensation reaction when we're talking about proteins. Now the same is true if we look at carbohydrates. So in carbohydrates, if we take 2 monosaccharides, glucose and fructose are both monosaccharides, they're simple sugars. And again, if we look at the functional groups, we've got a hydroxyl here and a hydroxyl there. And what will take place is we'll take the hydrogen from one, the OH from the other, water will be removed, and they are linked together into the disaccharide sucrose, and that's what we call table sugar. So the same thing that we saw with the proteins happens here in the carbohydrates, where water is being removed, and now we're starting a polymer. Our next macromolecule are lipids or fatty acids, and in this case we'll have a molecule of glycerol that has 3 hydroxyl groups. And what's going to happen here is we're going to take 3 fatty acids. This is why they're called triglycerides. We'll take 3 fatty acids, 1, 2, 3, and the acid part of that has the OH, and just as we've seen before, if we've got the OH and the H linked up, in each of these bonds, water will be removed. So for every triglyceride that's formed, 3 molecules of water are removed, and we end up with a triglyceride that looks like this. And so here is where that bond from the condensation reaction was made. And our final biomolecule is nucleic acids, and actually condensation reactions take place twice here. The first, when we form the individual monomer, the nucleotides. So we've got our nitrogen base, and we've got our sugar. And so to make the nucleotides, we have a condensation reaction that takes place right here to make our individual monomers of nucleic acid. Now, once we've got the nucleotides, then those individual nucleotides, just as we see over here, here's our OH and H, water will be removed, and we've just bonded two nucleotides together through something called a phosphodiester bond. It's still a condensation reaction, but that's a specific name for it. And if we look over here on the right, we can see here are several nucleotides that have been linked together to form a growing molecule of DNA or a nucleotide through the condensation reactions. So there's the 4 different types of macromolecules, 4 different groups, and they all are linked together with condensation reactions. Now, what happens with the reverse when we want to break them down through digestion? So the opposite happens is that instead of removing a molecule of water, here we've got to add a molecule of water, and the process is literally breaking water. It's called hydrolysis. And so what happens here is we add a water back in, that will break the bond, and we get back to our functional groups. We have the hydroxyl group and the hydrogen sticking off from there. So a couple specific examples. If we look here, here's the opposite. Here's sucrose. Here's our table sugar, our disaccharide right here. And if we add water, we can break this bond in here in the middle. It's called a glycosidic linkage for carbohydrates. But we break that, and the OH and the H are given back to the individual monosaccharides now, and we get back to glucose and fructose. So this is what happens in your body when you eat table sugar. You break it down, you digest it into the individual monosaccharides, and our body can use glucose much more efficiently than many other sugars. And just kind of quick recap, if you look at this, the top one is the dehydration synthesis or the condensation reaction. So here's where we will be linking individual monomers together with the removal of water. And then on the bottom, the opposite, hydrolysis. Here's where we're going to be breaking that bond with the addition of water to get back to our individual 2 monomers. And the last one I'll show is the same one, again, the cartoon version that we've seen before. The top one is showing the dehydration or condensation reaction, and the bottom one is showing the hydrolysis. So you can pause the video for a second if you want to spend a little bit more time with this. But that's the basics of macromolecules and how these polymers are going to be made. So next time we'll be looking at the individual categories of these molecules and the specifics of how these condensation reactions and hydrolysis work with those biomolecules. So thank you for your time.